everyone, I am Dr. Anjali Devi, Associate Professor, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. I am dealing with the course Enzyme Technology and presently I am discussing the topics included under Unit 3 of the syllabus. In the previous sessions, we have covered several topics included under this Unit 3. The topics that were covered are kinetics of single substrate reactions, estimation of Michaelis-Menten parameters, importance of Km, multi-substrate reaction mechanism and kinetics, turnover number, types of inhibitions, allosteric regulation of enzymes. And in today's session, we are going to discuss about product inhibition as well as deactivation kinetics. So, what is product inhibition? So, the end product inhibition, we are calling the end product inhibition as feedback inhibition also. And this is a form of negative inhibition. This is a negative feedback inhibition by which the metabolic pathway can be controlled. So, this feedback inhibition is very much important in order to control the metabolic reactions. In the end product inhibition, the final product that is obtained through series of reactions will be inhibiting the enzymes that are taking care of the earlier steps of the reactions. The product binds to the allosteric site of the enzyme. As I told you that your enzyme will be having active site, along with that your enzyme will also be having the allosteric site. This products will be going and binding to the allosteric site of the enzyme and they will be temporarily inactivating the enzyme. And what is the mode of inhibition? We can consider the mode of inhibition that is carried out by the product as non-competitive inhibition. As the enzyme can no longer functions, the reaction sequence is halted and the rate of product formation is decreased. I'll explain you in the subsequent slides. If the product levels builds up, for example, if there is a very increased levels of your product, then the product inhibits the reaction pathway and decreases the rate of further production of your product. If the product level drops in your body or if your product level drops, the reaction pathway will proceed unhindered and the rate of product formation will be increased subsequently. Let us understand the feedback inhibition with the help of an example. Just think A as a substrate. This enzyme 1 is acting upon the substrate and it is giving rise to B. And then this B is further converted into C with the help of another enzyme. This C is converted into D with the help of another enzyme and this D is your end product. For example, if there is a continuous production of D, there should be halt in the process so that the product concentration can be regulated. Then what's happening is this D will be going and it will be binding to the allosteric site of the enzyme, because of which this enzyme will be stopped acting upon the substrate molecule, thereby it will be stopping the further production of your product, that is nothing but your D. This halting process will be continued until your product concentration is regulated. Once, if your body requires the product D, then the inhibition will be stopped and the process will be carried out. The inhibition by the product D will be stopped and your reaction will again continue in order to produce more amount of product, which is nothing but D in this particular situation. Let me explain you about product inhibition with the help of another example. Let us consider isoleucine. This isoleucine is an essential amino acid. That means our body cannot synthesize this amino acid. We have to supplement this amino acid to our body through the diet. very important rich sources of this amino acid. Those are nothing but your eggs, seaweeds, fish, cheese, chicken, and lamb. In plants and bacteria, isoleucine is synthesized from threonine in 5-step reaction pathway. In the first step of the process, threonine is converted into an intermediate compound by an enzyme which is nothing but your threonine deaminase. This isoleucine can bind to the allostatic site of this particular enzyme, which is nothing but your threonine deaminase, and functions as a non-competitive inhibitor. As excess production of isoleucine inhibits further synthesis, it functions as an example of end product inhibition. Further, this feedback inhibition ensures that isoleucine production does not cannibalize available stocks of threonine. Moreover, if you consider the figure here, the starting material or the initial substrate for the production of your isoleucine is nothing but your threonine. Let us see this figure. Here we have considered threonine, which is your initial reactant, and many enzymes are involved in the process where this threonine is getting converted into isoleucine. Here, enzyme 1, 2, 3, 4, so 5 different enzymes are involved in the reaction where the threonine is getting converted into isoleucine. Here, what is happening is, in order to control the production of your isoleucine is going and binding to the allostatic site of your enzyme 1. Enzyme 1 is nothing but your threonine deaminase. Let us discuss few more examples which are related with your product inhibition. If at all you are considering the pathway, that is nothing but your heme synthesis, here the end product is nothing but your heme, which is inhibiting the initial enzyme, which is ALA synthase. In the case of cholesterol synthase, the end product is your cholesterol, which is going and binding to the enzyme, which is inhibiting the enzyme HMG-CoA reductase. In the case of glycolysis, glucose 6-phosphate is a product which is going and inhibiting the enzyme hexokinase. In the case of fatty acid synthesis, the end product acetyl-CoA is inhibiting the enzyme acetyl-CoA carboxylase. In the case of pyrimidine synthesis, your CTP is going and inhibiting the enzyme aspartate transcarbamylase. So these are the examples of product inhibition. This slide gives you a complete picture of various inhibition processes. For example, if you consider the competitive inhibition, here the structure of your inhibitor will be similar to the structure of your substrate. Similarly, in the case of non-competitive inhibitor, no structural similarity will be existing between the inhibitor and your substrate. In the case of allostatic inhibition, your inhibitor binds to the allostatic site of your enzyme. In the case of suicide inhibition, your inhibitor makes use of enzyme's own reaction mechanism to inactivate it. In case of feedback inhibition, the end product inhibits the enzyme that is involved in the metabolic pathway. Similarly, if you consider the factor of competition between your substrate for the active site, in the case of competitive inhibition, there will be competition between your substrate as well as your inhibitor for active site. In the case of non-competitive inhibition, there won't be any competition between your inhibitor and your substrate, as your inhibitor will be going and binding to the enzyme substrate complex rather than binding to the enzyme's active site. So in the case of irreversible inhibition, it causes conformational changes in the active site of the enzyme. If you consider the values of Km and Vmax, in the case of competitive inhibition, there will be increase in the Km value and no change in the Vmax. If you consider non-competitive inhibition, there won't be any change in the case of Km and Vmax will be gradually decreased. In case of irreversible inhibition, your Km won't be having decrease. Similarly, what are the various inhibitors that can be exemplified under competitive inhibition? Some drugs, sulfonamides, statins, all these are acting as inhibitors in the case of competitive inhibition. Similarly, heavy metal poisons, they will be acting as inhibitors in the case of non-competitive inhibition. So, this slide gives you an overall view of various types of inhibitions. Let us focus upon what is the importance of your enzyme inhibitions. The knowledge of enzyme inhibition is very much useful for understanding the regulation of enzyme activity within the living cell. It will be very much useful in elucidating the cellular metabolic pathways by causing accumulation of intermediates. For identification of catalytic or functional groups at the active site of the enzyme. Moreover, this provides information about substrate specificity of the enzyme. It will be very useful to study the mechanism of catalytic activity. The enzyme inhibitors have therapeutic applications. Some drugs useful in medicine appears to be a function by inhibiting certain enzymes. So, most of the drugs are competitive or suicide inhibitors. It is very important to know the significance of enzyme inhibition. Let us discuss about deactivation kinetics now. So, an important criteria when using enzymes in the technical process for material conversion or in analytical procedure is the stability of the enzyme during the process. As we have discussed previously that in order to use an enzyme in a certain application, in order to use your enzymes in certain industries, you need to have information about the stability of the enzyme under various temperature and pH and different pressure, etc. So, we need to study about the stability of the enzyme. It is very important criteria. The stability of the enzyme dictates the length of the time for which the enzyme is active under the reaction conditions. So, while studying the stability, we will be studying the activity of the enzyme, how much activity of the enzyme is persisting for how much time under various environmental conditions, at various temperature, at various pH, etc. So, you are going to study about the stability of the enzymes. Why we are going to study the stability of the enzyme? We need to know the information about how long your enzyme is stable at different conditions, different parameters. Depending upon the type of enzymes, its purity, enzyme pretreatment and reaction conditions, the enzyme activity can be changed detectably over the period of time ranging from minutes to years. So, some enzymes may be active only for few seconds. Some enzymes will be active for only for few minutes. Some enzymes are active for few hours. So, what are the factors on which the enzyme stability or enzyme activity depends upon? It depends upon the temperature, pH, etc. So, the stability of the enzyme depends upon the conditions in which your enzyme is prevailing. According to condition, it can be stable for few minutes or it may be stable for few years also. So, you need to study about the stability of the enzyme under various temperature, pH conditions. You have to note the time of stability. You have to note for how many hours or for how many months or for how many days your enzyme is stable and under that particular condition. The main factors affecting the enzyme stability are, as I told you, that very important factors that are affecting your enzyme stability or temperature, pH, concentration of your substrate and product, chemical effects such as inhibitors. The presence of inhibitors are also playing very important role. So, these are the very important factors. What are the very important factors that determines the stability of the enzyme? It's nothing but temperature, pH, pressure, concentration of your substrate and the concentration of your product. Along with that, the chemicals that are nothing but your inhibitors. As I told you that temperature is playing very important role. It is very important factor for enzyme activity. Temperature a dependency of the catalytic activity and stability of an enzyme. If the enzyme reaction follows a Michaelis-Menten reaction, then R is equal to R max into Cs divided by Ks plus Cs, where the maximum velocity of the reaction increases initially with temperature according to the Arrhenius equation. If you consider the same, according to Arrhenius equation, R max is equal to A into Ce exponential to minus Ea by RT, where A is the pre-exponential factor, Ce is the enzyme concentration, Ea is the activation energy, R is the universal gas constant, T is the temperature. At high temperatures, the conformation of your enzyme is altered because of the strong rotational forces and movement of your molecules, and this in turn has adverse effect on the ability of the enzyme to catalyze the reaction. I've told you that enzyme is very important factor that determines the activity of your enzyme. As your enzyme is your protein, temperature plays a key role on stability and the structure of your protein. At high temperatures, the conformation of your enzyme is altered because of the strong rotational forces and the movement of the molecules, and this in turn has adverse effect on the ability of the enzymes to catalyze the reaction. Every enzyme is active in its optimum temperature. I've told you that whenever you are controlling whenever you are considering an enzyme, and whenever you are testing or you are studying the activity of your enzyme under a range of temperatures, the enzyme will be active under certain range. That particular range in which your enzyme activity is highest, you are considering it as optimum temperature range. Every enzyme is active in its optimum temperature range. In this temperature, if you know you are exceeding the temperature slightly above your optimum temperature, there won't be drastic change in the conformation of your enzyme. But or you are deviating much from your optimum temperature, then there is every situation that your enzyme conformation may be changed. Every enzyme is active in its optimum temperature range. If this temperature is exceeded only minimally, then the conformational change will be reversible. That means very slight changes will be taking place. If you give back the normal temperature conditions, your protein will be regaining its original conformation. And the deactivation can be described thermodynamically. Irreversible deactivation processes are described by kinetic expressions. For example, if you kept your enzyme under drastic temperature conditions, there will be drastic change in the conformation of your protein. So you are considering that situation as deactivation. This enzyme activation will be falling under first order reaction. See here, this is your first order reaction. So we have seen that EN ED. Here En is the native enzyme. So enzyme in its native form, you have represented En as native form of enzyme, and Ed is the deactivated enzyme with the rate equation. dA by dt is equal to minus kDa, where A is the activity and kD is the rate of deactivation. Upon integrating this particular expression, A is equal to A naught into E to the power of minus kD into T. So your deactivation is highly depending upon the temperature. If you see this expression, kD is equal to kD naught into E to the power of minus EAD by RT. Let us consider this expression. K is equal to AD to the power of minus EA by RT. Here, K is representing the rate constant. EA is nothing but your activation energy. Here T is representing the temperature, which is measured in the units of Kelvin. And R is your gas constant. E is your mathematical quantity. If at all we are considering the deactivation kinetics and half-life, the thermal stability of your enzyme is quite variable. As I told you that the conformation of your protein is highly depending upon the temperature. As you are changing the temperature, definitely there will be change in the conformation. So the thermal stability of your enzyme is quite variable. Some enzymes lose their catalytic activity at lower temperatures, while others are stable at higher temperature also. And half-life is the time required for an enzyme to lose its half of its activity. So half-life is the time required for half the enzyme activity to be lost as a result of its deactivation. So that is represented as EA is equal to E naught by 2. So in turn, if you see, T is equal to T half. This is representing, T half is representing the half-life of your enzyme. So in today's session, we have discussed about your product inhibition, where the product, the excess product is going and it is binding to the allostatic site of the enzyme and it is halting the reaction where the product will be formed. It is halting the reaction by binding to the allostatic site of enzyme that is involved in the reaction. Along with that, we have studied about importance of your enzyme inhibition. Besides, we have also discussed about deactivation kinetics. I want you people to work out these questions in order to assess yourself how much you have understood from today's session. I want you people